Welcome back. We are starting with chapter 16 and we are starting with the mechanism of genetic variation. So in this chapter, we are going to see that how these bacteria that we were talking about in the last few chapters actually vary in their genetic inheritance because they cannot do any kind of sexual reproduction, but there must be some way that they do sustain some changes in their genetics. And we are going to talk about that in detail in this chapter. So the first thing that we are going to talk about is a mutation, which is a heritable changes in the genome. And we are going to see that how this happen in the bacteria. So by definition, the mutation is stable heritable changes in the sequence and the basis of the DNA. And the point mutation is the most common, what means that one base pair is changed by another base pair. That is what is called by the point mutation. And we are going to take a look at a couple of these, that how this happens. And the larger mutations are less common, and that involve insertion of bases, deletion of bases, sometimes inversion, duplication, and translocation of long stretches of nucleotide sequences. We are going to take a look at these as well. And the mutations can be spontaneous. As the bacteria are replicating, it can spontaneously happen. And there are some which can be induced. That means either the environmental factor or in the laboratory due to some chemicals, we can induce mutations. We are going to take a look at all of these. So the spontaneous mutation arise without any exposure to any external agents. And they may result in the errors in the DNA replication. DNA replication do have proofreading activity, and there are many different types of activity to maintain the fidelity or the integrity of the DNA, but there are still lots of possibilities of the mutations. One of the main reason for that is the chemistry by itself. And we are going to look at this, that this is what we know, what is known as due to base tautomerization. So the tautomerization result in the transition and transversion mutations. What we mean by these transition and transversion mutation is that when a purine is changed by another purine, that is called the transition. But when a purine is changed by another pyrimidine, that is called a transversal reaction. Now, that is a little bit more difficult because remember that purines have a double ring where the pyrimidines have a single ring. So changing a purine to purine or a pyrimidine to pyrimidine, which is a transition, is much easier because one single ring is going to be changed into and by another single ring of the base, or a double ring of the base is going to change into a double ring of the base. And this is okay to some extent to be accommodated into the DNA double helical structure. But if a single ring is going to change into a double ring, that is a little bit of problem because there are some steric issues of accommodating those structures to double rings in place of a single ring is much more difficult. So the transversions are less common. And this can happen due to insertion, deletion of the nucleotides, and also wrong base pair. And they may also result from the action of mobile genetic elements such as transposons. These are some of the other factors which can lead to spontaneous mutations. So the first, let's take a look at that how the base tautomerization can lead into the transition and the transversion mutations. To understand this, let's take a look at the different bases and the chemistry of these different bases. So we are only looking at the bases which are attached to the first carbon of the pentose sugar, deoxyribose sugar attached to the phosphate. We are only looking at the base. So if we are looking at a cytosine, here this cytosine or C is a normal condition, and you can see that in this position you have an NH. There is one hydrogen here, one hydrogen here. So this is an NH2. This form is an amine form of the cytosine, which is normal. On the other hand, what happens that it can change into another form, and this is what we call as tautomerization, that it can change into an imino form where you have this NH and this hydrogen is going to the adenine right here, bonding to the adenine, and there is no H in here. So this is changed from NH2 to NH. And this change can happen and that is a problem. The reason for that is the C base pairs with a G. It does not base pair with A. But if it is changed into this immunoform, here you can see that a C and A is base pairing. On the other hand, you can see right here, this is the normal form of adenine, which is base pairing with an immunoform, which is a changed form, AC base pair. This is a normal condition of adenine, which is an NH2 right here, which is changed into an immunoform here, just the opposite here. Instead of cytosine changing to immunoform, here the adenine is changing to the immunoform. It was NH2 here, and here it is an immunoform from amine form to immunoform, NH, just only NH. And this can base pair with a cytosine. So this is an AC base pair, 2 hydrogen bonds formed here, hydrogen bonds formed here. So either cytosine or adenine, if they change from their amine form to the immunoform, then they can cause these aberrant hydrogen bond formation, and this is transition in between them, and this cause problem, and this will lead to the mutation. On the other hand, there is another type of tautomerization which can lead to this problem. So if we are looking at this thymine right here, and this thymine do have a keto group right here. It's a keto group, it's a double bond with a C double bond O, so keto group. And if you look at this right here, this thymine, this is the enol form of the thymine, where there is a single bond and you have an OH. So this actually changed into an alcohol. This is an hydroxyl right here. And enol, how it is, this is an alkene and OL, all alcohol. So alkene, alcohol, that's how we call this an enol form. So this is the tautomeric form from the keto form to the enol form. And now if this enol form can form 3 hydrogen bonds with the guanine. So here you can see that T, instead of T and A base pairing, that is the normal, it is base pairing with G. T and G is base pairing due to this enol form. The same way, on the other hand, what can happen, that this is a normal condition of the guanine or normal guanine. And here you have a keto group right here, the same way that we have in thymine. You have a keto group, C double bond O. And here that keto group is changing to CO and 1 hydrogen is coming in, and this is the another tautomeric form or the enolic form of the guanine. If this happens, the guanine can base pair with the thymine as well. So you can see that in between A, T and G, no matter what, whether it is a thymine tautomeric form or guanine the tautomeric form, they can base pair in that way. Whereas in between C and G, whether it is a cytosine immunoform or an adenine immunoform, they can base pair. These are aberrant base pairing, and that this will cause problem. So this is what we call as the transition. And this is much more common type of mutation that we actually see. So here, how a spontaneous mutation is carried forward through the generations. So here we have a GC right here, and you can see that right here after the DNA replication, the 2 strand, here there is a GT, enol tautomeric form of the guanine is actually formed. So instead of a TA here, this is replaced by a G. Whereas GC, this is perfectly fine. So there is no problem with the first generation of this progeny on this side. Wild type, they will be the same as the parental. But here you have the GT, and now when the 2 strands separate, this form will have a C right here, and that is perfectly fine. But here, as you have a T, this was the mismatch, and this mismatch T is there will be the corresponding complementary base pair as A. So now you have a different base pair right here. So if we just read the DNA, here we are reading as ACG, here we are reading ACA. So here ACG, here ACG, but this is the mutant DNA. If this mutation is favorable, this will be a stable mutation into all the different progenies. So this is how the mutations can affect the replication in the process of replication. This is another much more drastic, which we call as an insertion or deletion, and they can lead to different types of mutation. So usually the insertion or the deletions happen when there is many bases one after another. It is just a random sequence where you have lots of same sequence. It could be just not A, it could be any of the 4 bases, And sometimes what happens that when the DNA polymerase is replicating these sequences, it can slip right here. So it's making these TTTs and it is slipping at this point. And if this happened, the slippage, see right here, the GT base pair make a loop out of this. And at that time, what happens, you can see easily that an extra T is incorporated in here, and this will be a mutation. On the other hand, you can see that this is the slippage in the parental strand as the slippage leading to a deletion. As this is being done, if there is a slippage here, you can easily see that there were two A's in here, and those two A's will not be copied into the strand, which is the daughter strand. So these two A's are deleted here. So here you have an insertion of a base, and here you have a deletion of these bases. So these insertion and deletions actually can lead to much more drastic changes in the DNA sequences. And we are going to talk about that in detail, what they can lead to. So here, another spontaneous mutation via formation of what we called as an apurinic site, or it could be apyrimidic site as well. So what happens here in the depurination can occur spontaneously. Say, for example, you have this sequence like TGGC, and suddenly from this side, this purine guanine right here just goes out. And this can happen spontaneously. So when you have the stand separation, your wild type sequence doesn't have any problem, no mutations here. In the first round of DNA replication, you have this, in this, you have this ACCG. So that will have an AACG. You do not have anything in here. So when the strands actually separate, here you have an TGC, and here you have an AACG. So here in the second round of replication, you are having an AACG or TTGC, whereas in the second round, this is the mutant, you have an AGCG. Here you will have an TCGC. So here it was TGGC, and here it will have a TCGC in the next round. So this will be an incorrect incorporation of a nucleotide generating a mutation. So these are some of the problem that can happen, which is the random depurination of a site in the nucleotides. So now we are looking at the induced mutation, and these are caused by agents that directly damage DNA. These are called some of them are called the base analogs, and these are structurally similar to normal bases, and mistakes occur when they're incorporated into the polynucleotide chain. And these are present in the environment by itself, or these could be some of the chemicals that the bacteria can be exposed to, and they can be incorporated into them for inducible mutations. There are many DNA modifying agents. They alter the base by causing it to mispair as well. We're going to see those. And the intercalating agents that distort the DNA to induce single nucleotide pair insertion and deletions. So let's take a look at a couple of them. So a very common two of these base analogs are 5-bromouracil and 2-aminopurine. These are the base analogs, and when they are incorporated into the DNA, the huge problem for this bromouracil is that they can form the tautomeric forms much more frequently than normal ATCGs. So much more tautomerization, and when those happen, that these uracil will base pair with other purines much more easily. So that's the bigger problem of these bromouracil or the aminopurine. Those are ethyl methylsulfonane. This is an alkylating agent that adds the ethyl group to guanine, hydroxylamine, hydroxylate cytosine. So you can see that many of these can modify the base, and they will mispair or mismatch with their corresponding complementary bases. On the other hand, you can see that acridine orange or proflavin, these are intercalating agents, so they will stack themselves in between the base pair. So you have these ATCGs, they will intercalate themselves in between them, and they can correspondingly, they can break the DNAs because of the steric problem. This acridine orange is interesting. This is a fluorescent dye that is being used for the localization of DNA in fluorescence microscopy. So this is a common intercalating agent, binds to the DNA, and we use this in fluorescence microscopy to identify where there is DNA in the cell. On the other hand, the physical, like ultraviolet light, promotes the pyrimidine dimer formation, which is in between the thymines usually. We're going to take a look at those. And X-rays can cause base deletion, single-strand breaks, cross-linking, chromosomal breaks. Much more drastic type of mutation can be caused by the X-rays. So these are the different mutagens which can cause what we called, as we talked about first, the spontaneous mutation, but presence of these mutagens can cause induced mutations. So here's two examples of an induced mutation. So here is a compound which is called methyl nitrosoguanidine. In the presence of this methyl nitrosoguanidine, a normal guanidine is methylated at the O6 position. So see that right here is a keto group, and this pairs normally with the cytosine. This is a G. Right here, it is there is a methyl group right here, and this will pair with the thymine. So you can see that just addition or modifying this base can change base pairing, and that can lead to mutations. So here we are looking at two thymines which can be in the nucleotide just next to each other. Here you have the sugar and the phosphate backbone, two thymine attached to the first carbon, two thymines, normal thymine. Now, in the presence of ultraviolet light, sometimes what happens, these two thymine next to each other, and only if they are in the DNA in a sequence next to each other, they will form a thymine dimer. So there is a bond formation in between these two, and this is what we call as a thymine dimer, and it's a problem because when DNA polymerase 3 is going to replicate this strand, it will pause or stall here. It cannot pass these two bases. So there will be a problem with these thymine dimer formation. So these can lead to mutations in the exposure to ultraviolet light. So the effect of mutation, we are going to see a couple of slides next to that. The wild type is the most prevalent form of the gene, and the forward mutation will change a wild type of mutation to a mutant form. On the other hand, these are some of the definition. Reversion of the mutation means a mutant phenotype is now changed to the wild type phenotype. Just a very simple way. Suppose there is a bacteria which bacterial color, the colony color of the bacteria is yellow. And now from the yellow color, the mutant form is red. So now in the reversion of the mutation, if that red colored mutant phenotype changes back to the yellow color, that will be a reversion of the mutation. On the other hand, suppressor mutation is a wild type phenotype is restored by a second mutation at a different site than the original mutation. So this is also another type. So just think about the same way that we have a site A and B. So if the A site changed to an A prime site, that changed the color of the bacteria from yellow to red. A changed to A prime, color changed from yellow to red. Now what happened, that there is another mutation where you have this A prime in the mutant form, which supposed to produce that red color from the yellow. But we have another change, whereas the first bacteria has a B, A and B. Now this second bacteria, along with the A prime, the B site changes from B to B prime. And the B prime will cause this bacteria to change the color back to yellow. So again, if we say that AB genotype of a bacteria, that is yellow in color. If it is A prime B, then it is red in color. But if it changes to A prime to B prime, then that B prime is the suppressor mutation, which is changing the A prime mutation from red to back to yellow. So that's your suppressor mutation. So let's take a look at the point mutation. We already talked about that. The point mutations is in a protein coding gene, can affect the protein structure in a variety of ways, are named according to if and how they change the encoded protein. And the most common types are silent, that the protein is not changed because there are some redundancy of the genetic code that we talked about that. We call the degeneracy of the code. Many of the codons, if just the third part of the codon is changed, it will still code for the same amino acid. Missense then this will be a different types of amino acid can be put into the protein. Nonsense means that it will put a stop codon in there, and the frame shift mutation will be usually by an insertion or deletion of a base, and that will completely change the reading frame downstream of that, and that will completely change the protein. So let's take a look a little bit of detail of what are those. So we'll spend some time on this type of mutations. So first we are looking at some of the forward mutations. We are changing to a wild type to a mutant form, and what they are. Suppose this is a DNA sequence, just random DNA sequence, where you have these amino acids coded by this DNA sequence: methionine, threonine, serine, proline, lysine, and glycine. So if there's a silent mutation, which we call as a base substitution, here you can see that the first base pair was first three letters or triplets are ATG, and then after that this was AC and A. So here it was ACC. Now that C is now changed to A to ACA. As I said, that the first two AC, these two nucleotide remain the same. If the third one is changed, most of the time due to the redundancy of the genetic code or the degeneracy of the genetic code, it will not cause a change in the protein. So the threonine remains here. This is a silent mutation. So the genetic code changed. So this is ACC, this is ACA, but this will not cause any mutation. This is a silent mutation, although the genotype changed, but the phenotype, there is no change. A missense mutation, on the other hand, is a base substitution as well. So you can see that right here, you have a sequence TCC, right here, TCC, that is now changed to TGC. And you can see that the serine is now changed by a cysteine, and this is a missense mutation because this is a different amino acid being put in there. Most of the times, if there is a single amino acid change, it may not alter the protein function, except if it is in a very crucial site, which is in the active site, or a hydrophilic or hydrophobic amino acid changed by other type. If a hydrophobic residue is changed by a hydrophobic residue, no problem. But if a hydrophilic residue is changed by a hydrophilic residue, usually not a problem. But if a hydrophobic residue is incorporated where there is a hydrophilic residue, that may cause the protein to fold differently and cause problem. On the other hand, in the nonsense mutation, you can see right here, GAAA's sequences here in this, instead of this AAA, you suddenly have a TAA. As soon as you have this TAA, remember that TAG, TAA, TGA, these three are the stop codons. So TAA will form UAA, which is a stop codon. So this triplet will code for a stop codon, and it will be the chain termination, or the polypeptide will stop right here. And most of the time, this will be a non-functional protein. And if this protein is required for the bacteria to survive, it will be lethal. If you are looking at the frame shift mutation, you can see that the insertion of this G will completely change the reading frame, and instead of the proline, lysine, glycine, this will be now alanine, glutamine, and arginine. So the rest of the protein will be completely changed, and that usually most of the time will cause a drastic mutation. So here we are looking at some reversion of the mutation by the base substitution. So in the forward reaction, here you have a methionine, threonine, and serine. In the forward reaction, it is changed to methionine to proline to serine, so due to this C change. And now this C is again changed into the reverse to A, and methionine, threonine, serine, the original protein is got back. So serine to cysteine, cysteine to serine, that is also another way that we got this sequence back. So here on the other hand is a polar amino acid, which is changed from serine, it is changed to proline. This is a non-polar amino acid, and usually these are drastic, as we said. But here, the serine is not replaced by serine, but it is replaced by a leucine, which is also a polar amino acid. And many of the time, if that is so, and if these amino acids are not at a very crucial site, like a catalytic site or anything like that, most probably the protein is going to be functional, if not 100%, to some extent. So this is what we call as a pseudo wild type, where a similar type of amino acid has been replaced. It is not the exact same amino acid in the reversion, but a similar type of amino acid. And this is also by the base substitution. On the other hand, we are looking at a suppressor mutation, and this is a frame shift of opposite sign, which are the intragenic suppressor by the insertion or deletion. So here we are looking at this sequence, methionine, threonine, serine, proline, lysine, glycine. In the forward mutation, we are seeing that here, due to the insertion of a G, right here, you will have an alanine, glutamine, and arginine. This was the sequence, which is due to the insertion and deletion. Now we have another suppressor mutation where you have a deletion. This base is deleted, and the deletion will cause to revert back to a methionine, threonine, proline, lysine, glycine. So here, just only in the middle portion, you have just a little bit difference. So instead of this serine, you have a proline here. This proline is still back in there, but you have an extra proline there instead of serine. And this is a suppressor mutation, which is another way that mutations can happen. Other than that, you can have an extragenic suppressor in the nonsense suppressor or physiological suppressor. Say, for an example, genes for the tyrosine tRNA undergoes a mutation in its anticodon region that enables to recognize aligned with the nonsense mutation, say, for example, UAG. Thus, an amino acid tyrosine is inserted at the mutant stop codon and translation continues. And that can give you an extra sized length of the polypeptide. So UAG is a stop codon. The same way it could be UGA or UAA. And if those anticodons of these are mutated in an amino acid anticodon, instead of a stop codon, the tRNA will insert an amino acid in that site, and the translation will continue until at the very end of the 3' untranslated region. That can lead to a longer length protein. Sometimes it may function, sometimes it may compromise function as well. The physiological suppressor is a defect in one chemical pathway is circumvented by another mutation, for example, that opens up another chemical pathway to the same product or permits more efficient uptake of a compound produced by a small quantities because of the original mutation. Suppose there is one bacteria which can form a vitamin, and this pathway is blocked by a physiological condition, and now it can form the vitamin by a different pathway. So these are some of the physiological suppressor that can happen. So we all talked about these things, so I'm skipping this. This is very self-explanatory. This just gives you another idea what is a silent, missense, nonsense, and frame shift mutation is all about. And now we are going to talk about the conditional mutation. This is expressed only under certain environmental condition. Think about that suppose there is a bacteria due to a mutation, it cannot grow at 37 degrees Celsius, but it can grow at 25 degrees Celsius. So we can tell that it is a temperature sensitive mutant, that in the cold condition that bacteria will grow, but it will not grow at a higher temperature or its normal condition. On the other hand, auxotropic mutants are unable to make essential macromolecule such as amino acid or a nucleotide, and has a conditional phenotype. So suppose one bacteria cannot make histidine, and it is an auxotropic mutant from histidine. So if the histidine amino acid is supplied to it from outside, it can grow. If without it, it will not be able to grow. Suppose one bacteria is auxotrop of cytosine. So if cytosine is provided into the media, then the bacteria will grow, but without the cytosine in the media, it will not be growing. So it's an auxotrop for cytosine. And the wild type strain from which it arose is called a prototroph. So a cytosine prototroph will be able to utilize cytosine, whereas a cytosine auxotroph will not be able to utilize or not be able to make cytosine. So it will need cytosine in the media to grow. So then we are looking at the mutation in the regulatory sequence. So say, for an example, constitutive lac operon mutants. We talked about the lac operon, and the lac operon in the normal condition is always turned off. So it is an inducible operon. That means a repressor constantly binds to its operator and it turns it off. Now there could be a mutation into the operator region of that lac operon, where the repressor can no longer bind to its operator sequence. So now that lac operon for that bacteria become a constitutive operon. That means that lac operon is now constantly on, and all the enzymes like a beta-galactosidase, permeases are produced all the time. Now, this is not the most favorable situation because you are making all these extra mRNAs translated into the proteins, which are not needed. It's a wastage of energy by the bacteria, but this can happen as well. On the other hand, the mutations in the tRNA on the ribosomal RNA genes, the protein synthesis can actually disrupt. So in the tRNA, what can happen that suppose the anticodon loop mutation can change the tRNAs from one kind of tRNA binding to another tRNA binding. So it will not read. So the mutation in the anticodon sequence can have very drastic effect in an organism because that specific tRNA, suppose I'm just giving an example, that's supposed to bring a histidine to the site. Now, due to an anticodon change, it is bringing glycine to the site. So every protein in that cell where a histidine is supposed to go in, it will bring a glycine instead. And that can have, or it will at least compete with the glycine tRNA and may insert the glycine instead of histidine in some of the areas. And that can lead to problems as well. And ribosomal RNA genes, if the mutation happen, it can cause problem to the ribosome. It can alter the rate of translation, or sometimes it could be a lethal mutation where the translation may not happen even at all. Or it can happen like a premature termination of the polypeptides. So that can happen. And many different types of compromised function can happen due to these mutations in these genes. So now after the mutations, we talked about what are the different types of mutation possible. And here we are now going into the practical idea that how we can isolate these mutants. So mutations are rare, 1 per 10 to the power 7 or 10 to the power 11 cells. And finding the mutants require a sensitive detection method or methods to increase the frequency of the mutations. But you have to remember that achieving those number of cells is not a huge problem in the sense, because think about that E. coli divides every 20 minutes. So the generations by which you can make those numbers are pretty achievable. And that's the reason in the bacterial world or in the viral world, just due to the extreme speed of the generations, these mutations are much more apparent than in the eukaryotic system. As I said before, in an animal cells divides every 20 to 24 hours, whereas within that period of time, E. coli will have way more number of generations, and the chances of getting the mutations are much more higher. So here we are looking at a mutant detection and how we can do this. So if a mutant do have a change, which is a visible change, say suppose a colony color change. Suppose it was creamy colored colony, now it makes a red colony. Don't take this example right here because this is just only for the purpose. But I'm saying, so let's not even talk about that. So suppose you have an yellow bacteria which now mutated into a blue colored bacteria, and that blue color is a mutation, and we can easily isolate that because there is a visible clue. But there are many strains which may have a mutation and you may not have any physical outcome of the mutation and you cannot detect. Say for an example, you have an oxotropic mutant, a particular bacteria that cannot make one amino acid and that needed to be supplied, that will be the oxotrop. And there is no apparent difference in between the wild type and the mutant. And how do you separate these two? So here we are looking at the power of the replica plating. So this is an E. coli cells with a mutagen such as nitrosoguanidine, and that caused probably the bacteria to mutate something. And we are thinking about that suppose there will be a lysine oxotroph that may grow out of it. So after adding the nitrosoguanidine, we are plating those bacteria onto a master plate where you have the complete media. You have all the types of amino acid in here. And now we are using a replica block with a sterilized velvet surface, and we press this on top of this master plate so that all the colonies will be right here on the plate. And then we gently touch our replica plate, which will also have the complete media right here. And here there is another media where there will be no lysine present. The amino acid lysine is absent in there. But we touch here first, we touch here second. All the colonies from here are transferred right here in the same impression. Now we are incubating those, and we are looking at that a particular strain that is growing in this media, but there is no growth on this media. So what we have, if we see that at the same place where you have a growth in this colony, but that colony is not growing in there, then this must be an auxotroph, because this cannot produce lysine. It is growing in the complete media where you have lysine, but it cannot grow in absence of lysine. And we can pick up that particular colony which is growing in the complete media but not growing in the lysine deficient media, and we can isolate a lysine media. So this makes it much easier rather than finding where is the mutant, because most of the mutants could be of the same color. They could be creamy colored here, they could be cream colored here, and it's almost difficult to find which is growing in here, which is not. And this is the replica plating, which makes it much easier to isolate the mutants. This is one of the process. So now we are going to discuss a test which is known as the Amy's test. And this was discovered by a Bruce Amis, and it's a very classic test to identify a potential carcinogen. So based on the observation, remember that most carcinogens are also mutagens. So the mutagens means that it will mutate the DNA, and that can lead to carcinogenicity. Now, why we do this test in the bacteria? Because it is much more difficult to identify the carcinogens, because animal experiments are difficult. But again, the mutagens may not be carcinogens unless it is described that it caused cancer in an animal model or in a primate or in the humans. We cannot designate as it is a carcinogen, but still this gives a good amount of preliminary idea that if it is a mutagen, it has the potential to be a carcinogen. So this process utilizes the technique which we call as a reversion of mutation. And for this process, the particular organism that is used is the Salmonella, and the Salmonella typhimurium is the specific serover that is used. And this particular organism is an auxotroph for histidine. So it cannot grow in absence of histidine. Histidine is an amino acid which is essential, and it's an auxotroph for that amino acid. And along with that, the test utilizes a particular culture of these Salmonella, which do have a much permeable cell membrane and the cell wall, so that the testing reagents that is being used, or the mutagens, can easily get inside the cell to have a mutagenic effect. So what we do, that we take this culture of the Salmonella and we mix it with the carcinogen minimal media and plate those in the minimal media plus a small amount of histidine. So we will plate this minimal media with a test mutagen and a small amount of histidine. So usually how the experiment is done, you have an agar plate, and on top of that agar plate, you spread that mutagen. Here into this plate, you have that the testing mutagen, and you spread that bacteria on top of that. Now on top of that, you put a little bit of molten agar, and that molten agar will contain a small amount of histidine. Here also, after plating the bacteria along with the mutagen, you will have a small amount of top agar, melted top agar, with a small amount of histidine. You are just layering that top layer, and you have a very trace amount of histidine, and you incubate them at 37. So the idea is that the top agar that you are putting on top of the bacteria will have an histidine which will allow these bacteria to grow only just for a few hours. And then the histidine will be completely depleted. Within that few hours, there will be no visible colony formation. So what happens that when you incubate longer, if you see that there are some colonies that are growing, then you can see that there are some spontaneous revertants. These were auxotroph for histidine, but some spontaneously, because here in this plate, you do not have the mutagen. These are the spontaneous revertants where the mutation has been reversed. Now these bacteria can utilize, they can make their own histidine, and that's why they are being growing here in this media without devoid of histidine. On the other hand, here we are seeing that we have added the test mutagen here. And if we see that there is the number of revertants are much more, significantly more compared to the spontaneous revertants, we can conclude that probably due to the mutagen present in here, the revertants actually happened, revertants appeared much more readily compared to the control here. And that's where potentially the test mutagen is a mutagen and probably a carcinogen. So this is the principle of the test using the Salmonella typhimurium histidine auxotrop and to the carcinogenicity test. This is known as the Ames test. So next we are looking at the DNA repair, processes of the DNA repair, and this is essential for the bacteria for the genome stability. So we are going to compare and contrast the proofreading, excision repair, base repair, direct repair, mismatch repair, recombinational repair, and also we are looking at an extreme scenario where an SOS response is described, where most of the bacterial DNA genome is severely damaged. So the proofreading is the correction of errors in base pairing made during the replication, and these errors are corrected by the DNA polymerase III. If you remember that, the DNA polymerase III makes the DNA from 5' to 3' direction, then it goes back to the 3' to 5' direction, opposite direction, scoots back, reads, and if there is an error, it's going to remove that and again start going in the 5', 3' direction. So it goes, backs up, goes, backs up, goes, backs up, and that's how it proofreads. Other DNA repair mechanism also exist, and one example is a mismatch repair. And this is a type of an excision repair where a piece of DNA is cut out where there is a problem. And this mismatch correction enzyme scans the newly synthesized DNA for mismatch pairs, and the mismatch pairs are removed, replaced by the DNA polymerase and the DNA ligase. So let's take a look at how this happens. So the first of this is used by a E. coli mismatch repair system and distinguish the old DNA strands from the new DNA strands. So the old DNA strand do have a methylation, and you can easily see that on the other hand, the newly synthesized strands are not methylated yet, because it takes, just after the synthesis, it takes a little time for them to get methylated. And the repair system cuts out the mismatch from the unmethylated strand. So how this actually happened in reality? So this is a stretch of DNA, and you can see that the repair complex have three enzymes, MUT-S, MUT-L, and MUT-H. So MUT-S scans the DNA, and suddenly it found there is a mismatch in correct base. There is a GT base pairing. MUT-S will stop there in this, and the MUT-L is associated with that, and the MUT-H is associated with the other strand, and the DNA strand is bent right here with this complex, and the MUT-H binds to a particular region which recognizes a methylation in the DNA. So this MUT-H protein of the complex recognizes that this top strand is methylated. See that this adenine is methylated. That's why this top strand is the parental strand. So it can distinguish that this strand is not methylated because it's newly synthesized, whereas the top strand is methylated. That's why it's a parental strand and synthesized for a while. So at this point, what will happen? That the MUT-H will make a cut to this non-methylated DNA, and then an exonuclease will chop up this strand. So here, the MUT-H cleavage site next to that methylated DNA, right here, due to this, and then this strand, which is the daughter strand, newly synthesized strand, is chopped out. Now DNA polymerase I will fill this gap, and then DNA ligase will make this correction. So this G is now mismatch repaired by this C. You can see that this is correctly replaced. So this is the taking the advantage of the DNA methylation. The MUT-SLH complex can make the mismatch repair. So Another process, which is called the excision repair, corrects the DNA that causes the distortion of the double helix. So we talked about these thymine dimers, where in between the two adjacent thymine, a double bond can form, and that will make a thymine dimer, and this thymine dimer will distort the DNA. So here, these complexes, which is UVR-AB complexes, and these UVR-AB complexes scans the DNA for any kind of distortion. When they find a distortion, this UVR-AB will bind to this distorted portion of the DNA, and if the damage is detected, the UVRAs are going to be released, and only the B stays there. And then the next enzyme, which is the UVRC, will come in and recruited by the UVRB, and it will make a nick adjacent to the area where there is a steric problem or there is a distortion in the DNA. It will make a nick cut, and this whole piece will be replaced out. And now what happens here, the DNA polymerase 1 will now fill the gap, and the DNA ligase will seal the gap, and this piece of DNA is repaired. So this is called the excision repair. You cut and then you repair that. You excise it and replace that sequence. So on the other hand, this is a base excision repair. So in the base excision repair, here you are making a length excision repair. So this is a little bit different, where there you have a distortion in the DNA and a stretch is repaired, whereas here you have just a single excision repair of a base. So here you have a mismatch, where is A and U base pair, and this is a wrong. Uracil should not be in the DNA. It should be in the RNA. So you have a wrong mismatch here. So the enzyme which is responsible for scanning those and finding those are known as DNA glycosylase. So the DNA glycosylase recognizes the abnormal base and cleaves the bond between the base and the sugar. So now you have an epiperimetic nucleotide right here, and the epiperimetic endonuclease recognizes the missing base and cleaves the DNA backbone on the 5' side of the missing base. So if you have this epiperimetic nucleotide right here, you have an A, but you do not have the pyrimidine here, it's going to make a nick, and the DNA polymerase 5' exonuclease activity will remove this region. It will remove this region, and then DNA polymerase 1 is going to fill the gap, and then DNA ligase is going to repair that gap. So this is another way that a base excision repair, if there is just only one base is damaged, this is how the repair is done. So excision repair is usually a distortion in the DNA, whereas the base excision repair, usually a wrong nucleotide in one place, is repaired by this process. On the other hand, there are some other process which is called a direct repair. One of the example of this direct repair is a photoreactivation. So thymine dimers are randomly formed when the bacteria are exposed to the ultraviolet light. And when the ultraviolet source is gone, then this is a little bit of extra that I'm saying that's not in your book, that when the ultraviolet source is gone, and the bacteria suddenly is in the dark, and if it happens, there is a particular enzyme which is called as a photolyase. This photolyase scans the DNA and bind to these thymine dimers only in the dark. And from the dark, when the bacteria again comes back to light, which means like daylight, not ultraviolet light, just the visible daylight, this photolyase will now become activated. It bound to the thymine dimers in the dark, but it did not do anything. It was just sitting there. But when it is photoreactivated in the presence of the light, it will just excise those two dimers out. The double bond will be broken and the normal bond will be restored. So this is known as a photoreactivation. This is for the thymine dimer repair. On the other hand, there are some alkylated bases. These are catalyzed by the alkyl transferase or methylguanine methyltransferases. These are the direct repair of some of the bases which could be modified in different chemical ways. So here is another way, is a recombinational repair. This corrects the DNA damage in both strands involved the recombination with an undamaged molecule. So in a rapidly dividing cell, another copy of the chromosome is often available, and that can do this. And there are many bacteria which are diploid, and the REC-A protein, or REC-A, is a recombinase, and this REC-A recombinase does that. So here is an example. We have a thymine dimer region gap, and as we have this gap, we have a distortion in the DNA. We have a gap in the daughter strand, which is the complementary strand being synthesized. So here what is happening in this process, that a region, this right here, this region is completely swapped with this. So this region will be excised out and it will go in this region. So when this happens, this is repaired, whereas this swap region, which is with this gap, so this portion is going in here and this portion is coming in here. So as this swap region doesn't have this will be repaired. DNA polymerase 1 will repair this gap, and DNA ligase will seal this gap. Whereas here you are seeing that as there will be a 2 A's the corresponding to the thymine. So this base, this will be broken. Now how this happens, even after this process, remember that there is the other proteins, which is right here to this one. Remember that this is the excision repair, where you have this UVR ABC system. So at this point, if we look at this, as there was a gap, the UVR ABC system will not be able to work due to this gap. But when this strand is continuous, UVR AB system can now scan this DNA, and it will find this thymine dimer, and it will replace that area, so the DNA damage is gone. So this is another way that the recombinational repair can happen. So the next type of problem that happens is the SOS response. So this is an inducible system, which is called a global control network, and this is used to repair excessive damage that halts the replication. So at this time, when this happens, there is a repressor which is called a LEX-A that is destroyed, and it activates over about 50 genes which are normally turned off. And the REC-A is a protein which initiates this recombination repair, and the REC-A is also a protease that destroys this LEX-A, so that as it destroys the LEX-A, which is the repressor, it will activate almost 50 more genes and increasing the production of excision repair enzymes. And also, along with that, there is another protein which we call as an SFI-A. This SFI-A is formed and it halts the cell division. So cell division is stopped almost like more than 40 minutes, and that time probably the repair damage can be taken care of. And other enzymes like DNA polymerase 4 and 5, they come in. The DNA polymerase 4 is known as a DIN-B, and this DNA polymerase 5 is also another enzyme, which is called the UMUCD. These are error-prone polymerases, and they synthesize the unrepaired DNA in the translation DNA synthesis process. So again, this is due to the lesion, like damage due to ultraviolet, X-ray, any kind of radiation, or mutagens. When you have an extensive break in the DNA, they try to rescue the DNA by this process, but still they are error prone, and that can lead to a lot of mutation as well. But this is the last process by which the bacteria try to save the DNA from completely being lethal to the organism. So the next portion that we are looking at is the microbes use the mechanisms other than mutation to create the genetic variability. And we are going to look at this horizontal gene transfer and the different processes of that. So remember that the bacteria doesn't have any sexual reproduction, so there is no vertical transfer. So there is no mixing of the parental genomes, and that's the reason that there is no crossover, there is no meiosis, and that's the reason that there is only horizontal gene transfer from one bacteria to the other. And all these mutations are subjected to selective pressure, and each mutant form that survives becomes an allele or a different form of that gene alternate form of that gene that we know from our basic definition of genetics. The recombination is the process in which one or more nucleic acids are rearranged and combined to form a new nucleotide sequence, which we call as a recombinant. And this is the process by which the bacteria produce more genetic variability. And this is what arises from what we call as the horizontal gene transfer from one bacteria to the other, or sometimes something is mediated by a virus or a bacteriophage, but from one to the other. It is not through the generations, but it could be from one group of bacteria to another group of bacteria, which could be related or could be completely unrelated. That's why we call it as a parallel or horizontal transfer of gene to make this genetic variation. So vertical gene transfer, here what we are talking about, transfer from the genes from the parents to the progeny. In eukaryotic reproduction, it's accompanied by the genetic recombination due to the crossing over between the sister chromatids. We talked about that, and the fusion of the gametes. This doesn't happen in the bacteria. That's why it has to be the horizontal gene transfer. And this differs from the vertical gene transfer of the genes from one dependent mature organism to another. And the stable recombinant has characteristic of the donor and the recipient. So you are getting from organism 1 to 2. The organism 2 will have all the characteristic of 2. In addition, if it gets a portion of the gene from 1, it will also have the characters of number 1. So this is actually an important factor in evolution of many species, expansion of the ecological niche. These bacteria can grow in the area where they were not been able to before. Increased virulence occurs by 3 mechanisms evolved by the bacteria to create the recombinants, and the genes can be transferred to the same or different species as well. So let's take a look at some of the basic ideas. Then we are going into the detail of that. Here are some definition. An exogenode is the DNA that is transferred to a recipient. So here we have a bacteria, we have a recipient chromosome, and you have a donor DNA. So DNA that is transferred to the DNA is an exogenode. Endogenode is the genome of the recipient. So here, if this is a recipient chromosome, this is an endogenode. So this is your exogenode, this is endogenode. So merozygote is the recipient cell that is temporally diploid as the result of a transfer process. So here, right here, this could be a condition where you have a merozygote where the cell is temporarily diploid due to the resultant transfer. So the homologous recombination is the most common form and usually involves a reciprocal exchange between a pair of DNA molecules with similar nucleotide sequences. And the REC-A recombinase proteins carry out this process. We talked about that many times. And a double-strand break occurs between the molecules, allowing exchange is mediated. So this is the most common type. On the other hand, there are some specific site-specific recombinations. This is important for the insertion of viral genome into the host chromosome. And there's only a small region of homology between the inserted genetic material and the host chromosome. And recombination occur at specific target site. And the process is also by the recombinase enzymes like REC-A. And 2 of the recombination used by the transposable elements to move from one place to the other DNA molecules. So we are going to take a detailed look about what we known as the transposable elements and what are those. So the next portion we are going to talk about that how these transposable elements move genes within and between the DNA molecules, and all of these are basically the recombination process, and this is the most common type to make genetic variations that we are going to talk about in detail. So the first thing, what we mean by transposable elements. The segments of DNA that move about in the genome is called a transposition. So earlier, a transposon used to be called as just like a jumping gene where they move. These are mobile genetic elements which move from one region to the other. But now by the transposons, what we mean that these are specific sequences in the genome which insert in multiple sites in multiple times, and that is definition of the transposable genes or transposons, and they have specific insertion sequences, and they contain extra genes, or which are actually known as the composite transposon. So let's take a look at these transposable elements. So the first one is an insertion sequence. This is the most common type. So the insertion sequence have a transposase gene, and all of these transposase genes will have, or all of these transposons will have an inverted repeat flanking the region, and then you will have a direct repeat in that orientation. So this is the most important part, that you have an inverted repeat and direct repeats. These are the signature portion that if all the transposable elements or insertion sequences will have these inverted repeats and the direct repeats. This just only contain a transposase. That's it. That's the insertion sequence. Whereas a composite transposon have an antibiotic resistant gene. This is the gene of interest needed to be going, or this is supposed to go in a different area of the plasmid, go different area of the chromosome. Along with that, you have a transposase gene right here, transposase gene, and you can see that the antibiotic resistant genes has inverted repeat next to the transposase, and the transposase will also have that inverted repeat with the direct repeats flanking the whole composite transposon. So this is a whole composite transposon. On the other hand, is an unit transposons, where you have a resolvase gene. This is your transposable gene, and you have only a transposase gene only on one side, not the other side like the composite transposons, and you have the flanking inverted repeat and the direct repeats. So this is the one that can go from one area to the other to the other. These are the different types of the transposable elements that is present. So how a simple transposition works, and we call this one a cut-paste transposition. The transposase catalyzes the excision of the transposable element, followed by the cleavage of a new target site and ligation into the site. So suppose here is a few hundred to several thousand base pair in length, and this is your inverted repeat. So the transposase recognizes the inverted repeats and cleaves both end of the transposase element. So it will cleave right here, it will cleave right here, and as it cleaves, now you have these inverted repeats next to the transposase right here. It's rounded up. Now the transposase carries the transposable element to a new site and cleaves the target unit at staggered site. It will make a cut right here. It will make a cut right here. So what you see, that it will be a staggered end right here. You will have a staggered end. So this was not here. So you will have only this portion and this portion. And this area, after making that staggered cut right here, this sequence will be inserted into this staggered cut sequence. So that is the most important part. Whatever you have cut, like ATGCT, that is right here, ATGCT, that's the portion right here, which is cut from here, AGCAT, AGCAT on this side. And so this and this was a base pairing. These two were base pairing initially. After cutting, they are now pushed apart. And in the middle portion right here, in the middle portion of this, because this was a staggered cut, in the middle portion, this transposable element is inserting its sequence. Now you have the DNA gap is filled. As the gap is filled, remember that you have this one strand of sequence right here is on this side, the complementary of that strand on this side, and this will be filled, this gap will be filled. So you will have a direct repeat here, you will have a direct repeat here. That's the reason that you have these inverted repeats on the end of inverted repeats and the end of these transposase or the unit transposons, and you will have the direct repeats at the each end. So this is a typical structure of a transposable element that we have seen, and this is how the direct repeats and the inverted repeats work. So here another process, the simple transposition. You have a cut, staggered end, and you just insert your transposable element in there, and that is done. This is just called cut paste. On the other hand, this is a replicative transposition, and here the original transposition remain in the parental site of the DNA, and the copy is inserted in the target DNA. So here you have the transposable elements which need to go into the target DNA. In the simple transposition, this will be excised out and it will be inserted into the target DNA. So the original DNA will not have this anymore. So cut, paste. Whereas here it is copy, then paste. So you will have the same copy of this one, and this will come out and will be inserted into the target DNA. So cut paste versus copy paste, which is the replicative transposition. So after that, we are looking at some other types of a horizontal gene transfer. A very common type that we just saw is the way of the transposable elements and recombination. And here we are looking at the bacterial conjugation, which is cell-to-cell contact. And we are going to talk about a lot of these different names: F+, F-, high-frequency recombination, fertility factor. So the first thing, that the bacterial plasmid is a small autonomously replicating DNA molecule, and it can exist independently from the host chromosome. And it also can integrate reversibly into the host chromosome, and in that case, it calls as the episomes. Now, this conjugative plasmid is a particular type of plasmid we call is an F plasmid. The bacteria which have the F plasmid are also known as the bacteria having a fertility factor, and we denote them as an F+. So an F+ bacteria is a bacteria with a fertility factor, or the F plasmid, and they can transfer copies of themselves to the other bacteria during the conjugation. So this is a property of this. So we are looking at this F plasmid, what you have, that they will have an origin of replication. And this origin of replication is known as an ORI-T, because we call this one as an origin of transfer. And you can see that this bacteria contain a lot of different genes, and these are called all TRA family of genes, and they encode these pyelin proteins. We are going to talk about that, what they are. They encode proteins of the type 4 secretion system. We are going to talk about what is type 4 secretion system. Encodes coupling protein. We are going to take a look at these, and also can encode relaxases, and we are going to talk about these in the conjugation process. So all the factors needed for the conjugation process are present in this F plasmid. So if a bacteria contain this F plasmid and designated an F+, that bacteria can transfer these F plasmid to another bacteria, and this is called the fertility factor, as I said. So how this F factor can be inserted? This F factor contains the formation of a sex pilus. So it attaches the F+ cells to the F- cells for the DNA transfer during the bacterial conjugation, and the F factors have the insertion sequences, or IS, and this assists in the plasmid integration. So how this happen, or the integration of the plasmid happen? So this right here, the integration sequence is present in the plasmid as well as in the bacterial chromosome. This is bacterial chromosome, and again, this will be a much larger circle, not linear. Insertion sequence where the recombination can take place, and this will be integrated. This plasmid will be integrated into the chromosome. If this happens, we call that this is an integrated F factor, and this can be copied into another and can be transferred to another bacteria. So if we are looking at this conjugation process right here, remember that the F factor contain information to formation of the sex pilus, and attach an F+ cell, means which has this F factor plasmid, to an F- cell for the transfer. So after transferring the F+ to the F-, this F- cell will become F+ as well. So Lederberg and Tatum demonstrated that a transfer of the genes between the bacteria depends upon the direct cell contact, and it's an unidirectional DNA transfer from donor to recipient. So this bacteria, suppose this is the donor and this is the recipient, so the F factor plasmid will be transferred from here to this bacteria. It's just a one-way traffic. So the first, they form a sex pilus, and this is a connection in between a donor and a recipient cell. And the sex pilus will now shrink, bringing them close together. And we are going to see that process. So we will come to that picture in a second. So let's take a look at how this fertility factor or F plus and F minus mating happen. So the copy of the F factor is transferred to the recipient and does not integrate into the host chromosome, and the donor genes usually not transferred. The F factor codes for sex pilus, and the type 4 secretion system makes the contact between the cells that moves across, and the plasmid is replicated by the rolling circle method. So let's take a look at what we mean by this. Right here, we are looking at the diagram of the F plus versus F minus mating. So here is a cell which has an F factor. This is the F factor plasmid, bacterial chromosome. And remember that by this means donor genes usually not transferred. The donor genes by this, what we mean by the genes from this bacterial chromosome are not transferred. Only the F factor is transferred. So the first here, you have an F minus recipient, which is a fertility factor minus, which does mean that it doesn't have the F factor plasmid. So the sex pilus is formed in between the donor and the F minus recipient. And the sex pilus contracts, bringing the cells together very close. And this forms what is called a T4S, or a secretory system of type 4 secretory system. And this type 4 secretory system is a conduit to transfer the DNA from this F plasmid to the other. And there are some coupling factors, and we're going to talk about that in a second. So at this point, what happens that there is a relaxosome, there is a particular protein that is responsible for replicating from this double-stranded DNA. It's going to make a copy of this DNA, of this F plasmid, by the rolling circle mechanism. This is just like thinking about that you are peeling off the DNA as it is being replicated. So this particular portion of the DNA that you can see, that will be attached to the coupling factor. And with the help of the coupling factor, this single-stranded DNA is going to be transferred into the new recipient cell, and the second double strand of the DNA will be synthesized. Now this bacteria becomes the F plus cell. So this F plasmid, a single strand of that is transferred. Now it becomes the double strand. Now this original F minus cell, which was a fertility factor negative cell, become a fertility factor positive cell. So this cell will now be able to do the same function again. So this is your F plus and F minus mating. So this is the conjugation machinery and how this rolling circle mechanism works. So here we are looking at the double-stranded DNA. And to do this, first, there is just a nick to the outside double-stranded DNA, the outside single strand. There is a break. And then the first strand is displaced. As the first strand is displaced, you can see that right here, you have this growing end because you have a 3' hydroxyl here. So the DNA is being synthesized right here in a 5' to 3' direction using this inner circle. So this inner circle is going around, and this is going around this to synthesize the 5' to 3' direction, the DNA. As this is synthesizing the DNA, this is going to get displaced, and this is the process is going to get displaced in this process. So you can see that as the whole 1 unit length of the DNA is displaced, you already have the double-stranded DNA is already being made. So in this process, what will happen that once you have this single strand, later the complementary strand, this is going to be transferred into the next. This is going to get coupled with these TRA-D coupling factor. And this portion, with the help of these ATPases, right here you have these TRA1 pylene. This ATPases will help this strand to push through this conduit to the next cell. So this portion is going to get over, and then there will be an excision, and this single-stranded DNA is going to get synthesized, or the complementary strand is going to synthesize upon this transfer strand. So you will have another full plasmid, F plasmid, in the recipient cell. And the original cell will have the double strand of the same F plasmid as well. So this is just is being shown that how this is the plasma membrane, peptidoglycan, periplasmic space. This is the LPS, so that you can easily understand that this is a gram-negative bacteria, and this is the conjugation machinery, how the DNA is transferred from the donor to the recipient. So here we are looking at another factor which we call as an HFR conjugation, which is a high frequency conjugation. So if you remember that here we talked about the donor genes are usually not transferred, means that if the donor contain the chromosome, the chromosomal genes are not transferred in F plus F minus mating. Whereas we are going to see that in the HFR conjugation, the donor genes are going to get transferred to the recipient cells. A complete copy of the F factor is usually not transferred, and the gene can transfer either in a clockwise, counterclockwise direction. So mainly these high frequency recombinations efficiency depend upon how much time usually the bacteria are sticking together, because we are going to come back to that. So let's first take a look. So here we are looking at an F plus cell. So this F plus cell has an F factor in the plasmid and the bacterial chromosome, which contain some of these specific. A lac plus means that this is a lactose utilization gene, and pro plus, this is a proline production gene. So this bacteria can utilize lactose and also can produce an amino acid called proline by itself. So in this bacteria, if this F factor, which is the plasmid, it can integrate itself into the chromosome. If this happens, this is known as a high frequency recombinant cell, or HFR cell. So this is different from the previous one. See that here, the chromosome, no matter what, all the time the chromosome remains separated from the F factor. So that's why this is called just only F plus F minus, whereas here you can see that when the F factor plasmid integrates itself to the chromosome, it becomes a high frequency recombinant. Now what happens? The same way that the two bacteria can contact themselves, and in between them you have this T4 secretory system, brings them apart, and the conduit is formed with the pyelin proteins and the TRA-D and the coupling factors. Now the F minus cell do have the chromosome, and see this chromosome does contain lactose utilization genes, cannot produce proline. So it's pro minus, lactose minus. So we are now transferring the high frequency recombinant chromosome. Remember that this red portion right here, this red portion, it will have that origin or ET, or the origin of transfer. So it can start making a single strand DNA after a nick, either in this direction, which is clockwise direction, or it can just go into the anti-clockwise direction. So it started from here and it just tries to go all the way up to here. And you can see that this red portion, it started from here, and then it got the lac plus at this point. This portion, the single-stranded DNA, is going through. Here you have the lac gene, which is from here. Then you have this pro plus gene, it's from here. It is going through the type 4 secretory system into the lac minus, pro minus, F minus cell. As this process is happening, if these two bacteria are attached to themselves for a short period of time, suppose at this point these two bacteria separate. So you can see that the lac plus got in here, but not the pro plus. Pro plus is still, proline is still in this cell. If the cells now separate, the bacteria only got the lac plus. So at this point, what will happen? This lac plus will get integrated into its genome, and now the bacteria, which was originally lac minus, can utilize lactose, so it has become lac positive. So this is an HFR F minus conjugation when there is a short period of time. On the other hand, suppose there is a longer period of time they are getting attached. Now you have time for even the pro plus or the proline manufacturing gene to transfer. So you have a lac plus, pro plus. So now the recipient is pro plus, lac plus as well. Here it was pro minus because there was not enough time for that gene to come in here, but here it is because it's lac plus and pro plus as well. So this is another scenario. So usually the bacterial chromosome, if the whole chromosome needed to be transferred, the two cells needed to be attached to themselves for more than 100 minutes. And that is a too long of a time. So most of the times, only the portion of the chromosome is transferred in the high frequency recombination. There is not enough time that the whole chromosome will be able to transfer to the other one. Theoretically, the whole chromosome can get transferred to the other bacteria, but in all practicality, that doesn't happen. Before that, the two bacteria go on their separate ways. So this is your high frequency recombination. So next one, we are looking at another extension of this same process, which we call as an F prime conjugation. This results when the F factor incorrectly leaves the host chromosome, and some F factor is left behind in the chromosome, and some host genes have been removed along with the F factor. And these genes can be transferred to a second host cell by conjugation. So by all means, F prime conjugation is a defective way to transfer the F factor. So here we are looking at the deintegration, including a part of the bacterial chromosome. So here you have the F factor right here, which is only the red portion. But due to when the F factor was leaving this, because remember that right here, the F factor, as it can integrate, it can come out from there as well. So both way traffic. It can be either separate or it can be integrated. So in this process, it is integrated, and this red factor needs to come out, and instead of being completely red, just only red, it brings a portion, this green portion of the chromosomal DNA along with it. So this is what is called as an F prime plasmid, where you have a little bit of sequence from the chromosome to the F factor plasmid. Now it is trying to transfer the material to another F minus recipient from the F plus donor. This is not an F plus donor, this is an F prime donor. And in this process, what is happening, that it is transferring a portion of the genes to this second one, which it derived from its own chromosome. So this is the idea, and these genes can be transferred to a second host cell by the conjugation. And this is the overall process. So the next portion is the bacterial transformation, and the bacteria can uptake DNA from the environment as well. So in this process, we are looking at the bacterial transformation, where the fragment of the DNA fragments, double-stranded fragment, they can be taken into the DNA, but usually when it's taken up, only a single strand of DNA goes into the cell. There are two things can happen. The bacterial DNA can undergo homologous recombination and can be integrated into the chromosome, or it could be degraded by the Cas9, CRISPR-Cas9 system, or any other process, which is the unsuccessful transformation. On the other hand, a double-stranded DNA plasmid can also go into the cell and stay separate from the chromosome as well. So in this process, we are looking at, there are some particular bacterial genus which are very good at this. One of these is a Streptococcus pneumoniae. They can do this particular uptake of DNA from the environment or transform very, very well. And to do this process, a particular condition of the cell needed to be met, and we call this as the cells being competent. So in a growing cell, only about 10-15% of the cell are in a condition or in a physiological condition where we call them that they are in a competent condition. What we mean by a competent condition of the cell, a competent cell can only take up the DNA, does have the machinery to take up the DNA into the cell. Not all the cells are competent. And in the laboratory, we can make the cells competent by chemical treatment to take up the DNA, or sometimes by using an electrical pulses, we can make the cells competent transiently as well to take up DNA as well. So here we are looking at a bacterial chromosome inside a bacteria, which doesn't have a lactose utilization gene. And here is a lactose utilization gene from the environment. And the bacterial extracellular endonucleases cuts the DNA into a smaller fragment. And after cutting it, just only one strand is degraded and a single strand passes inside. And after coming inside, some homologous recombination can make the DNA integrate into the chromosome, forms an heteroduplex of the DNA, of the new DNA template, create a lac plus gene, and now this transformed cell contain the lac plus or lac utilization genes are in this bacteria, which came inside by the bacterial transformation. So in this process, the DNA uptake, this is a type 2 secretion system, and it is a pretty complicated system. So here first we are looking at a gram-positive bacteria. You have the plasma membrane and a thick peptidoglycan layer, and you can see these COM proteins have this long pilus which attaches the double-stranded DNA and brings it to this COM and the endonucleases, and these endonucleases chew up one of the strand of the double-stranded DNA, make it a single strand, and that single-stranded DNA with these proteins pass into the cytoplasm of the cell. So this is the process in the gram-positive bacteria. Whereas in the gram-negative bacteria, you have this different protein, which is the pyelin protein, different types of pyelin protein. You have the plasma membrane, then you have the peptidoglycan layer and the periplasmic space. Here you have the outer lipopolysaccharide layer, and here you have also the COM A, E, all these different types of protein. Right here, the probably COM E, which is not really very well characterized, will make the double-stranded DNA to a single-stranded DNA by chewing up one strand, and it will deliver the strand inside. And this is the way that the bacterial DNA can be uptaken by this process of transformation. And again, there are many areas that the scientists are working on and not completely or clearly known that exactly the mechanism how this happens. So the last part that what we are looking at is the transduction of the viruses. And here the bacterial DNA can be transferred by the phage, which are the viruses which infect the bacteria or bacteriophages. So we are going to take a look at this. So this is the transfer of bacterial genes by viruses or bacteriophages can carry out a lytic cycle. The host cell is destroyed, or a viral DNA integrates into the host genome, becoming a latent prophage. So let's explain this. So these bacteriophages can come in two different forms. So the first type of form, we call them as a virulent phages. So the virulent phages go into the cell. They will make lots of copies of the phages or the virus, and the cell will burst, and all these cells will undergo lysis. This is called a lytic cycle. So the virulent bacteriophages will cause the lytic cycle. Host cell is destroyed and the new viruses will come out. So this is one way these bacteriophages reproduce. On the other type, what happens that the bacteriophage is called as a temperate phage that goes into the cell and it integrates its viral DNA into the host or the bacterial genome. And this is called a latent prophage. So this virus or the prophage will remain inside the bacterial chromosome for an indefinite period of time. And at one time, this latent prophage can again excise itself out from the bacterial chromosome and then make a lot of copies of these prophages. So new bacteriophages will be made out of these prophage genes. They will be packaged inside that, and it will go into the lytic cycle. So it will be virulent. So some of these temperate phages can integrate their own genome being in a latent prophage, and that is the temperate phage nature. But these temperate phages sometimes can turn into virulent phages or undergo that process where this type of process, which is known as a lysogeny, will be converted into the lytic cycle. So when a DNA is integrated into the host genome as a latent prophage, this is known as a lysogenic cycle. In the lysogenic cycle, the bacteria is not killed immediately, but the lysogenic cycle can go or induced into a lytic cycle when the prophage excises itself out into a productive production of the bacteriophages. So let's take a look at the generalized transduction. This is the lytic cycle of the virulent phages. So here you have a bacteria chromosome, which does have a histidine and a lysine positive. Histidine positive, that means that this bacteria can produce histidine and also can produce lysine. These are the two amino acids. Phage DNA is injected into the bacteria. Once the phage DNA comes inside the bacteria, it triggers some specific process where the host chromosomal DNA or the bacterial chromosomal DNA is chopped up into pieces. All the machineries of the bacterial cell is engaged in making only the phage DNA. And this phage DNA is going to transcribe into the mRNAs. Those mRNAs are going to made into the phage proteins, and they are going to be packaged inside the phage. So the phage DNA is red in color. So more red DNAs will be made in the cell. They will be packaged inside the phage. But in this process, these fragmented DNA from the cell, if they are of the same length of this red DNA, which is the phage DNA, and it's just by chance and just by mistake, some of this bacterial DNA can be packaged into a virus head right here by mistake. And this is a transducing phage with the host DNA, and this is just an aberrant mistake. So now the lytic cycle will lyse the cell, so all these bacteriophages will come out, and this particular bacteriophage, which is transduced with the host DNA, will come out and infect another new cell, and it will inject its own DNA, and this packaged the histidine plus genotype into its head. So now it is incorporating the histidine genotype into a cell where you do not have an histidine positive genotype. It's a histidine minus. So after this process of transduction, this cell now become histidine positive, or it can produce the histidine, and this is known as the generalized transduction. And the generalized transduction works in a way that just by mistake, a wrong packaging of bacterial DNA into the bacteriophage. Whereas a specialized transduction is carried out only by the temperate phages that have established a lysogeny. Only a portion of the bacterial genome is transferred and occurs when the prophage is incorrectly excised. So here you have a lysogenic cell with a prophage. So the prophage sequence is in the red and is integrated into the chromosome. And here what happens, it includes some of the genes of the bacterial genes. So right here is a condition where the excision of the prophage DNA happens. In the process of this excision of the prophage DNA, which is supposed to be this, it carries out, or it takes a few of portion of the bacterial chromosome with it. So think about that. The prophage is the red DNA. It is excising it out. In the process of excising it out, it, instead of the red DNA, complete red DNA, it now contains a portion of the blue DNA. So this is not a complete prophage what originally came in, but it is a little bit different. So now this red-blue DNA, they will form the genome of the new bacteriophage. The proteins are making into the capsids and the tails and everything part of the bacteriophage. And these hybrid DNA, which is a portion of the prophage and the portion of the host cell chromosome, will be packaged into these bacteriophage. This will go into a new cell. And once this DNA is injected to the new cell, that can have an homologous recombination with the new cell, and it can produce a new type of genotype. This is another way. So in this process, here is the bacterial genome, the chromosome. Here is the incoming bacteriophage DNA that can be integrated just the same way as we saw, and it can remain as the prophage as well. On the other hand, there could be an homologous recombination as well that can happen. So these are some specialized transduction. So here we are looking at a transduction in E. coli with this phage lambda. And phage lambda do have some specific sequence, which is called the attachment sites, or ATT sequences. So these ATT sequences are recombination sequences. So in E. coli, we have these genes, which are, say, GAL, so for galactose, and BIO+ for the biotin. So it can produce biotin and it can utilize galactose. So these two Gal plus and Bio plus sequences are flanked by an ADT sequence. So by this recombination, the lambda phage can integrate itself. It can integrate by the homologous recombination in between this Gal plus and Bio sites. So this is an integrated prophage. Now what happens that there are two ways that at one fine day that this particular phage can excise itself out of the bacterial chromosome, and this is your specialized transduction, and this is happening at the ATT site. So when this excision happening at that time, you can see in this process, you have in the normal condition, you have right here is a lambda phage, and the lambda phage is excised out. So there is no problem with this. It was an integrated lambda phage. It excises itself out. So this can produce a lytic cycle, and all the lambda phage will be the same type that came into the cell. But sometimes you can have an error in the excision. So here the DNA is wound like this, and while the excision is happening, it is not in between the ATT sites. So as it is not happening in between the ATT sites, that's right here, it is happening somewhere else. A portion of the Gal here, but it could be a portion of either a Gal or a Bio plus gene can be transferred or moved into the phage DNA. So this is an error-prone excision. Now what happens that this particular phage can go into a new cell, and it will cause an homologous recombination. And if this new cell was Gal negative, it can receive the Gal positive gene from there. So this is here to a Gal, and again, remember carefully, here the bacteriophage delivered the Gal utilization gene to a Gal negative genotype. So this is how a specific viral transduction works, where the virus is delivering a gene of interest to the bacteria. So here we are looking at that how the evolution in action actually happened and the development of antibiotic resistant bacteria. And this is the long big picture outcome of this. And we are going to look at this R plasmid, which conferred these antibiotic resistance. So how this immunity genes work in the bacteria, it's not really an immunity gene. These are called the resistant genes that exist in nature to protect the antibiotic-produced microbe from their own antibiotics. Because if a bacteria is producing an antibiotic, and if that antibiotic is detrimental to itself, it's going to commit suicide. So there are some particular genes that produce some specific enzymes to degrade those antibiotics produced by this bacteria. But sometimes what happens that these immunity gene or resistant genes of the bacteria can be transferred to a different genus or a different species, and that's how they move in nature. And this is the horizontal gene transferred immunity genes from antibiotic producers to non-producing microbes. So the producers are the ones which contain the resistant genes to save themselves, but they can move to other bacteria, and these bacteria, if they are disease causing, now have these resistant genes, and when we treat them, try to treat them with an antibiotic, they will be resistant against them. So these resistant genes can be found on bacterial chromosome, they can be found on bacterial plasmid, transposons, other mobile genetic element, and found on many different areas. So let's take a look at the chromosomal genes. The resistant forms are rare, and spontaneous mutation usually result in a change of a drug target. They can be present on composite transposons, contain a genes for antibiotic resistant. Some have multiple resistant genes and be inserted and reinserted, and can move rapidly between plasmids and through a bacterial population. So very quickly, resistance to antibiotic actually can rise. So here we are looking at the R plasmid, and can be transferred by the cells by conjugation, transduction, or transformation, and can carry multiple resistant genes. So if we are looking at this right here, you can see that it's resistance to kanamycin, ampicillin, and sulfonilamide. There are so many, chloramphenicol, there are so many resistance. And interestingly, if you carefully look at that, it does have a TN4 transposon. Inside that TN4 transposon, you have a TN4 transposable element as well. So if we look at that TN3, you have a transposase. This is the trip A. Then you have a resolvase, trip R. Then you have an beta-lactamase right here, molecular weight about 20 kilodalton. What you are seeing is a transcription direction. So the transcription direction of the transposase is towards this. Transcription direction against of the resolvase are towards this. And each one of them have a 38 base inverted repeat, and you have a target site flanking repeat in there. So what does this beta-lactamase do? Beta-lactamase, again, we are going to talk about that in a separate chapter in detail. Beta-lactamase confer resistance to all penicillin group of antibiotics. This breaks down a particular beta-lactam ring in the penicillin group of antibiotics and render them ineffective. So sometimes the beta-lactamase is also called as the penicillinases. So you can easily see that the transposons rich areas, that this can move from one bacteria to the other by all different types of horizontal gene transfer, which is conjugation, transduction, transformation. Then they can insert themselves into the chromosome constantly or from one plasmid to the other. So all the different types of integrations are possible so that they can be conferred to new different species and group of bacteria very, very efficiently. So this is just an example that how these antibiotic resistant genes move into the nature. So that's pretty much all. We looked at the genetic variation of the microbes, fundamentally different from the sexual reproduction methods. And we also saw that how the jumpstart of genetic variants happen. Looking at the evolution, we looked at the bacteria. Viruses have very short generation time and frequently only one copy of their genome and how mutations affect them. Looking at a much more bigger picture, and we are going to talk about that how these changes affect the overall antibiotic resistance and a problem of the common warts. And we are going to talk about those in much detail in a particular chapter. So that will be pretty much the end of your chapter 16.